Today we're going to have a discussion about the antigenic composition of bacteria. And the lecture is really also related to the immune system in the sense of we're going to look at different structures of bacteria in relationships to how they enable recognition by the immune system. And the possibilities are immense in terms of the different ways that bacteria can present themselves in terms of being recognized by the immune system. So we're not going to cover everything. We're just going to really cover a survey of kind of the most extreme ways in which bacteria are recognized. And then as we go through this actual survey of the different kinds of bacteria, we will review and cover the antigens and the different parts of the bacteria that serve as antigens when we get to that lecture. So, how can we describe bacteria in relationship to their structures that serve as antigens? So we're going to originally talk about the antibody-antigen relationship or interaction and how antibodies identify antigens. And again, I refer you to several of the immune system lectures where that is covered in much more detail. We're also going to talk about the actual adaptations that bacteria have in relationship to which of those adaptations also serve as antigens. We're going to talk about different parts of the cell wall of the bacteria and how they function as antigens. We're going to talk about the bacterial capsules and how they serve as antigens, also flagella. And then we're going to talk about just within a species how we can observe and detect antigenic variation, and then we're going to talk about how this variation is enabling us to develop vaccines. So the antigen-antibody interaction, it can be, now really the antigen can be basically any molecule, and its unique shape is recognized by the adaptive immune system, most notably the T cells and the B cells. And once the unique shape of the antigen is recognized, then the immune system launches a series of different cytokines and different cells and different mechanisms in which that pathogen can be eradicated. So generally, these B and T cells are especially modified in relationship to recognition of the antigen. So some examples of antigens can include proteins, carbohydrates, cells, whole cells, cellular structures, enzymes, particles, all kinds of things can actually serve as antigens. And a really good example of that is, for instance, allergies, that people develop allergies to things like pollen or bee stings or peanuts or various things in the environment, cat fur or cat dander. And all of these things actually serve as antigens that are presented to the immune system, and then a autoimmune process develops where we have a reaction against that antigen. And in some cases, in the case of asthma, for instance, that's maladaptive. But what we're going to talk about today is really what parts of the bacteria can actually serve as antigens, and how can those antigens best be served by eliciting an immune response. And you know, again, the bacteria are very clever, and they've evolved different mechanisms for being able to evade immune detection. So we'll cover those in today's lecture as well. So, to ensure survival, now remember, bacteria live in quite a different numbers and kinds of environments. They live in environments with oxygen. They live in environments without oxygen. They can live as spores right in the earth for years. They can live in all different kinds of tissue. And so in terms of being able to live in that wide a range of environments, bacteria have evolved very specialized mechanisms, and in particular adaptations to the human host environment. Now, these adaptations can also serve or also function as antigens. And then, of course, these antigens are then recognized by the human host, and then an immune attack is launched against those antigens. Now, some antigens, some other bacterial antigens, are actually the pathogenic agent in which disease is caused. So, for instance, we have some antigens that are basically toxins that are secreted by the bacteria, and the human host has developed antibodies against these toxins. So that's another example of how different aspects or parts of bacteria can serve as antigens. Now, remember, there's a very important principle about what enables the immune system to recognize an antigen. And so the antigen really has to be a big enough size, and there are various criteria for determining that. But if the antigen is not large enough, then it can escape detection. The other attribute of antigens that enable detection is how different the antigen is from the host. So both viruses and bacteria have developed adaptations that enable escape from detection because they actually mimic the human host. And in the case of viruses, as we discussed, the viruses actually can take on the host cell membrane and escape detection that way. And the bacteria does other sorts of things that also enable the bacteria to potentially evade detection by the immune system. Also, the antigens need to be complex in shape. So if they, again, resemble a cell, for instance, maybe they won't be detectable. But if, on the other hand, they're very complex in shape, then that facilitates a stronger or a better, a more refined level of recognition. So what are some of these bacterial surface components that can be antigenic? Well, in our bacteria morphology lecture and in our cell wall discussion, we talked about various structures such as the pili, right, and the flagella, the Fimbriae. Now remember, the Fimbriae are these spines that enable the bacteria to attach to tissue. The flagella, of course, is a mobility device, and the pili are, remember, those bridges between cells that enable transmission of genetic material. We also have capsules, and remember, capsules surround the bacteria, and they can serve certainly as an antigenic structure that is recognized by the immune system. Remember the peptidoglycan layer, remember gram-positive and gram-negative. Remember gram-positive, that peptidoglycan layer is right on the outside of the cell. There are these S proteins, and these S proteins are what's known as sensing proteins, and these can actually detect variations in the environment and can even sometimes signal the cell to undergo various somatic cell changes and to better adapt to the environment. The M protein and the lipid A protein, and we learned about these in earlier lectures on bacterial structure and bacterial adaptation. So here, this is the surface of Bacillus anthracis, and you can actually see the P for peptidoglycan layer here. We have S for the sensing proteins, and then we have the capsule out here. So any one of these structures could certainly serve as antigens for detection by the immune system. So here is an example of the Fimbriae. Right now, Neisseria gonorrhea grows inside epithelial cells of the reproductive tract. So the bacteria use these fimbriae to actually anchor themselves at the tissue, and this outer membrane protein called OPA can actually facilitate entry of the Neisseria gonorrhea bacteria into the epithelial cells. So after attachment, the host cell actually takes in the bacteria through endocytosis. Okay, so here you can see the fimbriae. Here's an example, Neisseria gonorrhea, and it also has a capsule. Now, the fimbriae can also be antigenic, right? And so how are they antigenic? So we can have secretory antibodies, IgA, that can block bacterial colonization by blocking attachment to other bacteria by the fimbriae. Other circulating antibodies can target bacteria with fimbriae and opsonize the bacterial cells for phagocytosis. So different parts even of the same bacteria can serve as, multiple different antigens that target those immune cells to the same bacteria. So in the case of Neisseria gonorrhea, we have the fimbriae that we can utilize as an antigen. And we also have the capsule as well. Okay. So what about the capsule? Let's talk about the capsule. Now, the capsule serves several different kinds of function in the bacteria. So these capsules are generally composed of polysaccharides. Now, again, what is a polysaccharide? It's a very long chain carbohydrate. It's a complex carbohydrate. And these capsules can contain antigenic components. So, but also, the capsule can also serve as a major determinant of virulence. So what is virulence? Virulence is the capacity to cause disease. So if a pathogen is highly virulent, it's highly pathogenic in relationship to quickly and easily causing disease. And also the disease can be more severe in terms of its level of infection. So antibodies to the capsule, in this case, also can function to neutralize the virulence. So for instance, if you get a capsule surrounded by antibodies, you can create an agglutination reaction, which essentially neutralizes the virulence, because the disease, in this case pneumonia, is really elicited and caused by the capsule, by the antigenicity of the capsule creates this huge immune reaction which causes these symptoms of excess mucus and fever and so forth. So what other kinds of defense mechanisms do these capsules have? And can these capsules also serve as antigens? Okay, well, we talked about pneumonia. Well, what about some other ones? We also talked about anthrax and pyogenes. So these are the two, the pneumonia and the pyogenes are both streptococci. And all three of these have capsules. So the pneumonia capsule actually directly evades phagocytosis. Now, what is that? That's the engulfing of the bacteria by an immune cell. Anthraxis, remember, is a pretty serious disease, and that capsule protects bacteria from complement-mediated lysis. So remember that complement occurs in the innate immune system in the beginning of an infection. So having the capsule around the anthrax bacteria actually protect it from being lysed or, destroyed by complement. Now, pyogenes is actually, that capsule is very clever because that capsule actually is made of this hyaluronic acid, which is a component found in human connective tissue. So basically, the capsule of the pyogenes mimics human connective tissue. And so in that case, it can escape detection by the immune system as well. However, these three examples also, these capsules also serve as the antigen of that particular bacteria. So that actual structure, the actual capsule, serves as the recognition molecule for eliciting the immune reaction. And that's very important because remember, if we have that agglutination reaction, we can also neutralize the effects of that bacteria. So now here's another example. Okay, so this example, and really the purpose of this lecture is really to present several examples that can present kind of concepts and enable your understanding of the myriad possibilities that we have in relationship to different aspects of bacteria that can serve as antigens. So here, the motility, this Vibrio cholera, which is a very serious bacteria, is the flagella itself is antigenic. So the antibodies actually recognize the flagella, which to me is fascinating, right? And so what happens is when the antibodies surround the flagella, they can actually cause the Vibrio to, for instance, stop movement. And the way in which these cholera infect the intestinal tract is they actually burrow, they actually swim. If you can imagine, they actually burrow their way into these intestinal epithelial cells. So if you can agglutinize or agglutinate or immobilize the bacteria by directly acting on the flagella, that's a very elegant way in which you can potentially eradicate that pathogen. But again, with cholera, it's a very rapid onset. Boy, those bacteria, once they get in, they immediately burrow into those intestinal epithelial cells, and then potentially they can escape detection too. Okay, so really, but you can imagine they also, by doing that, they avoid elimination by peristalsis, and they also avoid the digestive enzymes. So these are other types of examples of antigenic variation in bacteria, but also in protozoas and also in fungi. So you can see there's 3 or 4 examples of different bacteria, and what's shown in this table are different proteins that actually serve as antigens. So here with this meningitis, the pili itself actually, in this case, serves as the antigen. But we also have another antigen. We have the OPA protein, and that can serve as an antigen too. So the interesting thing about these bacteria is they don't just have one antigen. They can actually have more than one. So in that case, it makes it easier to recognize them and also eradicate them as well. So here is a discussion and a description about antigenic variation in the same species over time. Now we know, for instance, with influenza, we can get quite a bit of variation over time. But what about bacteria? Well, the interesting thing about bacteria is that if they can change their antigenic structures, then potentially over time they can actually escape destruction by the immune system. So this is a really important concept. It's a concept that isn't necessarily restricted to viruses, that bacteria can also, as we know, recombine themselves into new strains. And if they do, are the new strains different enough from the old strains so that they too can escape destruction by the immune system. So in this case, this is again Streptococcus pneumoniae. And now it's a bit challenging to read these figures, but I'll do my best to explain them. So in this case, you can see that there are green triangles and red triangles and blue ones. And those are meant to represent variation. And you can see now these are two different geographic sites. So you can see that these two sets of plates, I guess you can call them, are pretty similar geographically. And going this way, they are also pretty similar in relationship to time. Now, they're not exactly similar, but they're similar enough. And then in this plot here, in this graph here, you can see that there have been 3 different strains of Streptococci pneumoniae that were collected in a particular hospital. And on this axis, this is percentage of invasive disease isolates. You can see that there isn't that much difference over time. And these, these are, approximately, 5 to 10 year intervals. And these different times, these samples were collected, and basically the strains were pretty much the same. So in this case, there wasn't much antigenic variation over time. Now, recently, in the last several years, we have developed vaccines against the pneumonia bacteria. And so, for instance, if you get a vaccine if you get vaccinated for pneumonia, chances are the vaccine is going to last quite a while because the strains don't change. Now, again, that's very different from influenza, for instance, where you need to get revaccinated in some cases every single year because of that antigenic change. All right, so what about this example? So here is an example. This is meningitis. This is Neisseria meningitis. Bacterial meningitis is a very serious disease, and we will discuss it in the bacterial survey lectures. But here so we have the same thing. We have variation by geography, and then we have variation by time. Now you can see in this example, there's variation by geography, and there's variation by time. So you can see, and this is a global serogroup distribution, and you can see that there are differences in the strains. So there is substantial between host diversity that is changing over time and by geographic region. So within the host, there isn't much diversity. So once you get that meningitis, boy, that's going to be with you and it's not going to change. But in terms of over time, and obviously in different countries, you are going to see differences. So here, if you look at Asia, for instance, Asia is pretty much has the same strains. Now, the United States and Europe have some variation, and Africa has quite extreme variation from the other countries. South America is a lot like Europe. Okay, and so is Australia. So you can see there's some things in common, but there's also variation as well. And in comparison to the pneumonia, there's quite a bit more variation. Now, how can this antigenicity be utilized for vaccinations? Now, we will have a separate discussion about vaccinations, but just for the purposes of this discussion today, we're going to indicate or just provide one example here. And this example is to actually employ a part of the bacteria that can serve as the antigen. And again, this part of the bacteria has to be consistently expressed and be constant. And so in this case, what happened was we took, and this is an example. So here we can take a polysaccharide coating, right? Maybe even part of the peptidoglycan layer. And we can actually insert it into a carrier protein. And that carrier protein is a protein that's recognized by the immune system. So the immune system will take it in, and then it will actually develop an immune reaction against the novel part of the protein. And we will develop an immune recognition of this polysaccharide portion. And so then when we administer the vaccine, that creates a very, very subclinical, right, infection where we develop both B cells and IgM. We develop antibodies that have a memory. And remember, T cells have memory too. So we develop a subset of T cells that have been especially designed. The T cell receptor has been especially designed to recognize this part of the bacteria. And so then we have these subsets of memory antibodies, and we have memory T cells. And the next time the individual is infected with this particular bacteria, the immune system will launch an attack immediately because it has already recognized this bacteria as being a pathogen. So that's how parts of a bacterial structure can be utilized in relationship to developing vaccines, for instance. But, as we can see in this lecture today, we saw that they were capsules can serve as antigenic determinants. We saw flagella. We saw potentially parts of the bacterial cell wall, pili, also the Fimbriae. Remember the lipid A and the toxins? The toxins can serve as antigens. And there are various different aspects of bacteria that can certainly serve as antigens. So it's important to have an understanding about that the immune system has basically evolved different ways to be able to detect infection by bacteria. Now remember, there are some bacteria that are friendly as well, and all of those are actually covered in various lectures in the immune system. So the first question is, what is the underlying determinant of antigenic variation? Now this is a very important part because we have two systems here. We have the bacterial system and then we have the human host. Well, in general, what is the mechanism by which we can change, right, the structure of a bacteria? Now we know, for instance, that bacteria recombine into new strains. So what part of the bacteria engages in recombination? Well, guess what? It's the DNA. Okay. So in terms of the bacteria and the human host, they can actually co-evolve, right, together. So what we can do is we can have the bacteria developing into various strains over time. And what enables that to occur is recombination that occurs through genetic change or through genetic exchange, right? Now, in terms of the human host, we can also have coevolution of the human host. However, unfortunately, that takes many, many, many more years, right? But theoretically, we can actually have inheritance. We can have, for instance, we had a very serious bacterial global infection, like the bubonic plague, for instance. And we wiped out a huge section of the population. the individuals that were left behind were potentially the ones who could survive, that the immune system figured out a way to eradicate that pathogen. And so those survivors then become a new gene pool. So we can evolve over time through, through adaptation and inheritance, again, of genetic material. And, also through selective reproduction and survival. So basically, the underlying determinant of antigenic variation is DNA, okay? That when the environment changes, at least in the case of bacteria, the bacteria change along with it. And when they change, they potentially recreate themselves into a new strain, and then they can become antigenic, right? They can become newly, a new emergent species that then need to be recognized again. So strains can develop enough difference from the original strain that the immune system will need to relearn the new structural variation. So that's very important. All right, so the second example, what are some criteria that are required for immune recognition of the antigens? Now remember, there were three. So one, it has to be the antigen needs to be large enough. Okay. It needs to be complex. Complex in shape, or sufficiently complex in shape, and it needs to be sufficiently different from the human host. So, thank you so much for listening to this very important lecture today. And again, it ties in with a number of other lectures. It ties in with our morphology, bacterial morphology lecture. It ties in with our bacterial adaptation to the human host. It also ties in with several lectures on the immune system. And really, the purpose of today's lecture is really to talk about the myriad possibilities, the myriad ways that the human host can use structures from the bacteria to enable recognition. So that concludes our lecture for today. Thank you so much for visiting educator.com.